she passed away from the flu on Friday. She went to sleep and never woke up. She's 36 years old. She had the flu, was in her lungs, started going to pneumonia, took some NyQuil, and never woke up again. Isn't that crazy? One of my close friends, she has a child and was sleeping. Parents came home and checked on her, had her visit, come by, and she was just goners. So yeah, I was like, the flu? So it's like we have flu and we have, if you don't get it managed, flu. Yeah.
So it's because she just didn't get it managed or not?
Let it go too far. She probably should have been in the ER like 2 days prior. It turned into pneumonia, didn't get that in check, went to sleep, and I'm sure there might be other complications you're not aware of, obviously. So they didn't do an autopsy, but it was just, yeah. So that's what I'm saying. You're careful. It sounds like the flu and the cold, right? No big deal, but in the wrong body, right? Maybe some medical complications, not managing it, it can go south pretty fast. Okay. So in adaptive immunity, everything we did was innate before in chapter 16. Okay, innate is ready there to target anything that comes your way. Okay, it's a general attack system. Okay, then it's like throwing a grenade at something. We're not that specific about it, so we'll kill whatever's coming our way. Okay, the adaptive immunity is the snipers. Okay, targeted attacks for going after one thing. Okay. You only have adaptive immunity whether if you had that infection yourself and you generated antibodies against it, you responded to it, but you had to get it through an infection, or you get it through vaccination, okay? That you introduce that antigen into your system, maybe a toxoid, a less virulent form of the pathogen, and then mounted an immune response against it that way. So natural infection or through vaccination. You always have two responses in that system. You have a primary and a secondary, okay? Your primary is gonna be a very short-lived response, okay? The second time you encounter that antigen, you're gonna generate memory cells and you're gonna have a long-term response that the antibodies stay in your system longer. Your titer is higher and they stay circulating in your system for weeks instead of just a few days, okay? Because your body says, oh, I remember you. I don't have to do the whole setup again. I have memory cells that can be activated and then come and attack. Okay. We have two types of immunity, humoral immunity and cell-mediated immunity. Humoral immunity is referring to B lymphocytes and their ability to produce antibodies. So B lymphocytes will secrete antibodies that circulate through your blood, and they will target antigens that are outside of cells, okay? So our B cells are lymphocytes, okay? Remember that big circle nucleus with a little thin rim of cytoplasm around it? Okay, we have B and T lymphocytes. So the minute they've encountered an antigen or been presented with an antigen, they can make antibodies. The name comes from Bursa fabricus. This is only in birds, and it's their version of a B cell factory. It's a lymphatic nodule that's only in birds, and this is where B cells were first discovered, and then hence the Bursa, that's where the B lymphocyte comes from. Okay, cellular immunity or cell-mediated immunity is when T lymphocytes are produced and then activated. So they come from the bone marrow and then finish the maturation process in the thymus. They are going to recognize antigens that are intracellular, so such as a virus, intracellular bacteria, chlamydia, something like that. And then that cell says, hey, this thing's taken over inside of me. Let me take a little piece of it and show you what it is. I'll present it to you, and then the T cell attacks. So it's anything that's intracellular, and then the T cells will attack those pathogens inside. Okay? So once a T cell, each T cell has a receptor called a TCR or T cell receptor. Those are what's going to bind the antigen that the cell that has an internal infection. This cell with an internal infection says, hey, this is what I'm dealing with. Let me put it on the outside of my cell. The T cell binds to that, and once you have that interaction, the T cell's activated. The T cell will secrete a lot of messengers called cytokines. and those cytokines will activate cells downstream. So we'll talk about those different pathways. So every cell in your body comes from what's called a pluripotent stem cell. Those stem cells are in your bone marrow. Okay, within your bone marrow, they turn into B lymphocytes. B lymphocytes stay there and mature there. Okay, T lymphocytes will leave the bone marrow and finish their maturation process in the thymus. Okay, you make about as many T cells as you're ever going to need by puberty, and then your thymus starts to degenerate. And so you might have, as you age, then you, with your T lymphocytes, you have less abilities or less attacks with those. Okay, both the B cells and T cells, you're going to find them once they finish their maturation process. They're going to migrate to your spleen. Your spleen filters all your blood for pathogens. They're going to migrate to all of your lymph nodes. Your lymph nodes have B cells and T cells. They will be found in your tonsils. Your tonsils are there for whatever you're going to eat to filter and encounter a pathogen. You guys remember what it's called in your small intestine? The lymphatic nodules in your small intestine, in the ileum, Peyer's patches. Okay, Peyer's patches, tonsils, lymph nodes, spleen. Those are all lymphatic tissues, and they all have B lymphocytes and T lymphocytes there ready to be activated. So cellular immunity, T cells. Humoral immunity is B cells. So cellular immunity is going to use T cells to attack intracellular pathogens. Humoral immunity is going to use antibodies to attack pathogens or antigens outside of the cell, circulating in tissues and fluid in your bloodstream, but it'll be outside cells. When T lymphocytes, for example, are activated, you're going to have different types of cytokines produced. Cytokine just means cell messenger, okay? So you have, that's the broad term, you have several types of cytokines. Some of the more popular ones that you see coming up over and over again, interleukin, so in between white blood cells, okay? They're messengers between maybe a T lymphocyte, a helper cell, to a B cell to cause antibody production. Chemokines, those are the ones that are attracting phagocytes to a site of infection, that chemotaxis process. Interferons, we talked about interferon alpha and beta, are produced when a cell is infected with a virus. They cause the next door neighbor cell to produce antiviral proteins. Tumor necrosis factor, that gets produced in all autoimmune diseases. You'll have higher levels of that. And then hemopoietic cytokines, when I taught you the hematopoiesis chart, we talked about the myeloid stem cell line and the lymphoid stem cell line. Myeloid goes to erythrocytes and our granulocytes. The myeloid stem cell line goes to B lymphocytes, T lymphocytes, monocytes, and natural killer cells. So you have those two lines. But there's multiple stages before you get to those final cells. So there's a lot of what we call cytokines that promote immature forms of those cells to go to the mature form, like an erythroblast before you go to an erythrocyte, a monoblast before it becomes a monocyte. So that's what causes the maturation of those stages. All right, so I keep using the word antigen. I think you guys have an overall idea of what we're talking about, but simply it is a substance that causes the production of antibodies, okay? Antibodies will bind to an antigen. Antigens can be a lot of things, but they're usually going to be some type of polysaccharide or protein on the surface of a microbe, of a fungus, of a virus, of a bacteria, anything, an algae, okay? So antibodies have these sections of them, maybe a few sequences of amino acids, a few sugars within that polysaccharide, called an epitope. An epitope is what's going to bind to the antibody specifically, okay? So I can so if I call Kayla the antigen, okay, and I'm the antibody, I could say her shoulder is an epitope. My interactions, I'm only gonna bind to a specific area in her shoulder, okay? That's the antigen determining region. There are some antigens that are too small to evoke an immune response, and they have to be conjugated with a larger molecule. So you'll put them with carrier molecules. In vaccines, it's called, if it's too small of a protein or sugar, they're put with an adjuvant, and the adjuvant helps get an immune response stimulated. Otherwise, if it was just that one protein or that one little stretch of sugars, it wouldn't stimulate. So when you're looking at a vaccine and you see the term adjuvant come along, that's what it's for. It's to help it to happen. Okay. The interaction between an antigen and an antibody is very, very specific. Okay. So if I say I have 3 potential antigens on the surface of the cell, I have that square one, I have the triangle, and I have the semicircle, right? Okay. The triangle antigen can only bind to an antibody that has the same shape. So of all my antibodies, only antibody B with a triangle orientation can bind to the triangle antigen. It's a highly, highly specific interaction. So we have millions and millions and millions of antibodies in our system. You have one for any antigen that you could ever encounter in your lifetime, okay? So the variety is huge and the specificity is very tight. So they're not just gonna indiscriminately bind to random antigens. Okay, we talked about haptens. Okay, so we're looking at antibodies. The fancier name is immunoglobulin. Okay, they're proteins within serum, they're globulin proteins within our serum. So we use the general term antibodies. The antibodies can bind X number of antigens. That X number of antigens is called valence. Okay? If it's a bivalent antibody, it can bind 2 antigens. There are different classes of antibodies, and the valence will change with that. So our antibodies, let me take you to the picture so I can describe this. The antibodies are made out of 4 polypeptide chains. You have what's called those long chains are heavy chains. Okay, they're polypeptides with more amino acids in them, so they're heavy. You have 2 light chains in blue. Okay. The bottom portion of this here that you see C is called your constant region. Bottom portion of the light chain is also the constant region. You have 5 types of constant regions: G, A, M, E, and T. GAME. 5 classes of antibodies: IgG, IgA, IgM, IgE, IgD. So GAMED is a good acronym. Okay. So we only have 5 classes of antibodies or constant regions. So how does that account for the millions of antigens that you could potentially interact? So you have this variable region, okay, and the variable region changes when you're looking at the DNA level. The variable region in our genes, the amount of switching of nucleotides that happens is just mind-boggling to be able to express a protein, a variable chain that is unique to an epitope. Okay, so we have our variable region on the light and the variable on the heavy. Those both will contact the epitope on the antigen surface. Okay. The chains are held together by disulfide bonds. So remember way back in the beginning of this class, right? Disulfide bonds are from the amino acid cysteine. So you're gonna have these cysteine residues that are anchoring those particular chains together. Okay. This region here, the FC region, stands for crystallizable fragment. The crystallizable fragment, those 2 constant regions, those are often bound to receptors on the surface of certain white cells. So let's say a mast cell or a basophil, for example, they will have receptors specific for IgE. When IgE binds to the receptor, it causes histamine to be released. So we're going to get into those type of dynamics. But those will, the constant region, the FC region will bind to receptors on certain leukocytes.
I'm sorry, it's called crystallized what?
Crystallizable fragment. Oh. Okay, so let's work through our 5 classes. Our IgG, so we call a monomer. It's going to be bivalent. It's got that standard Y shape. Okay. This is the most abundant of our antibodies. You have 80% in your serum. This is a really important one because it's the only one that crosses the placenta. It's the one that's gonna protect the baby. It's when you're breastfeeding, even if you choose not to breastfeed long-term, they always tell you that you have to feed for the first few days to get the colostrum produced. It's not milk, it's just yellow stuff that comes out of your breast. And that is rich in IgG antibodies. So even if you don't breastfeed, you need to for the first few days, weeks, just so that baby can get their initial immune system established, and then after that, you know, personal choice after that. Okay, IgG can also coat a pathogen and make it more appealing to a phagocyte. That's opsonization. We talked about that last week. That complement can activate opsonization. Opsonization is the coating of antibodies. IgG is one of the classic ones in this. IgM, let me show you what it looks like real quick. I will come back to this slide. Okay. IgM is this guy right here. Looks like that guy. Sorry, that was bad. Okay, so it's 5 immunoglobulins joined by disulfide bonds at the FC region. Okay, so that increases its valence to 10. It can bind 10 antigens at the same time. Okay. IgM, it's a lower in abundance, so we're talking about 6% of our antibodies. It circulates in your blood at all times. It doesn't get into tissues, it doesn't get into lymph, stays in your bloodstream. Okay. Because it has 10 spots, it's really good at clumping. So when we do a agglutination test, we will use IgM because that's what causes the visible clumps. Let's say if we're doing an RPR for syphilis, if we're doing an agglutination blood type test, those would be all classic agglutination tests where we use IgM. Okay. When you're having your primary response, so you have a primary and your secondary. Your primary is short-lived. IgM is the first antibody out or produced the first time you encounter an antigen. So let's say I've never had mononucleosis. I'll pick a little more random one, okay? So if I've never had mono, first time I come across someone mono, okay, then I'm gonna have IgM produced. Second time I come around mono, we'll live through that hopefully, okay? Come around second time with mono, I'm gonna have different antibodies produced at a higher level in the secondary response. And my body hopefully remembers what the first one was. Okay. IgA, it is potentially two antibodies that will be bound together by what's called a J chain. Another name for it is the secretory chain. Okay. So when it's in serum, when it's in blood, it's in the monomer form. Then if it gets into mucous membranes and secretions, usually your GI tract, okay, also vagina, any type of secretory mucous membranes, it will be in the dimer form, so it'll have a valence of 4, combined 4 different antigens.
You said it was 2 antibodies put together?
2 antibodies, they'll be joined. I'll show you the picture in just a second, yeah. So they're joined, let's take the long part of the Y's, the stems, they're joined together. So these guys, you're gonna see them in mucous membranes. They're gonna be in your tears, they'll be in your saliva, and there's another one that will be in the breast milk that the baby needs to get to establish their immune system. Okay, it's there. So when we're looking at the modes of invasion by Salmonella and Shigella, remember they get into those intestinal columnar cells, they're causing the membrane ruffling, right? They're trying to get just past that first layer of mucus membrane of columnar epithelium. So IgA is there to bind it and block it from ever attaching and potentially causing membrane ruffling and getting inside. So if you have IgA, it should be there to block it and interfere.
So IgG and IgA are important for breast milk antibody production?
Yeah,
or
for
is it a
fetal.
combination of, like, all of it?
So IgG will be able to cross the placenta, so that's the initial antibody. It will also cross in breast milk, and so will IgA. Yeah. Okay. IgD is one of the least abundant. They were at 0.02%, so much smaller number. It is a serum antibody, and it's a very similar structure to IgG, so valence is what?
2.
2. Okay. It's a little bit larger antibody. The FC region is longer on IgG. The main function that's known on it is bound to B lymphocytes. And when I use the term B cell receptor, which you're going to hear me use a lot in a few minutes, the B cell receptor is an IgG antibody that's bound to the surface of B lymphocytes, and it's there waiting to bind antigen. That's really the only solid function that's been determined so far. Our last one, our least abundant 0.002% in serum antibodies is IgE. Okay, IgE is going to dock on the surface of mast cells and basophils. Remember I said there can be receptors on mast cells and basophils that bind to the FC portion of IgE. When IgE locks and loads onto a mast cell or a basophil, it stimulates the granules to release histamine, okay? So then the histamine causes maybe an allergic response. It can also come and bind to, part of it will bind to, let's say, a big old parasitic worm, okay, like maybe Enterobius or a whipworm or something like that. IgE will bind to that worm all over, coat it, but it doesn't, like one phagocyte can't engulf a whole worm. So what it does is it brings a lot of phagocytes in, a lot of destructive cells, and they all work together to try to like destroy this worm in chunks. So we'll get back, it's called ADCC. That's one of the last things we'll talk about. Okay. So just like eosinophils target parasites, you'll have a higher level of eosinophils when you have a parasitic infection. IgE, the E's, are going to be abundant when you're fighting parasitic worms. This is a very good summary chart, okay? You should know this thing right here for a good compare and contrast. You should be able to know how to draw an antibody, and label it correctly, explain its function, explain its valence, and the differences between the 5 classes, okay? So this chart, I really like this one for a summary. Okay, changing gears a little bit to talk about something, a major histocompatibility complex, okay? We have these on every one of our cells in our body, okay? So your body has MHC genes that code for MHC receptors. You have 2 classes, MHC1s and MHC2s, okay? Every cell in your body has MHC1 receptors, except for erythrocytes. The only one, because they don't have a nucleus. Every other type in your body, and that marks you as self, okay? If you have MHC1, this is your unique sequence or setup of MHC1s, means this cell belongs to Bethany, okay? The second type is we have MHC2s. They're on the surface of what we call antigen-presenting cells. An antigen-presenting cell, the cell that can engulf a bacteria, so that means it's phagocytic, right? It can break that cell up inside, break up the pathogen. It takes a little piece of that pathogen and sticks it on the surface, an MHC2 receptor. And it says here, it's like if I went inside myself, took out a little piece of E. coli that's troubling me, okay? And I said, here's a little part of it, here's an antigen, and I present that in my MHC2 receptor. So antigen presenting cells will engulf bacteria, phagocytose them, take a small portion of it and present it to our T cells and our B cells. So now we're going to talk about activation of B cells and T cells and how we select which cells are going to be expanded. So I'm going to go here to this picture. I like this one a little bit better. Okay, so we have our B lymphocyte. On the surface of the B lymphocyte, we have this antibody structure, right? Okay, this would be our B cell receptor, aka IgD. Okay. So our B cell receptor, remember, antibodies have a specific, the epitope, Okay, they're only bind to one epitope, they have an antigen determining region. So they have that triangle shape. So that means of all the potential extracellular antigens, okay, combined antigens are outside of cells. Okay, we have square shape, we have circle shape, so it's only gonna bind to those squares. So it says, okay, I have got you, I'm gonna internalize you and break you up into several fragments. Okay. Then those fragments then get presented on MHC2, the yellow guys here, those are MHC2 receptors. So it takes it and it puts that little fragment of the square on the MHC2 receptor. So now the cell, the B cell, is presenting the little fragment. So it's an antigen presenting cell. Okay. Once it presents it, same thing here, here's a little triangle that is presenting. A T helper cell, okay, has a T, put a big arrow right here to that orange guy, that's called a T cell receptor. That T cell receptor is going to bind to the antigen that's presented by the MHC2 on the B cell. Once that interaction happens, that activates a T helper cell, and that T helper cell will secrete cytokines. Those cytokines come back and activate a B cell, that same B cell, and say, hey, B cell, okay, I recognize what you presented to me, I'm gonna activate you, and now you're gonna make two types of cells. You're gonna make a plasma cell and a memory cell. The plasma cells, they have a high abundance of Golgi apparatus and rough ER because they're gonna be a factory for secreting antibodies. So plasma cells secrete antibodies that are specific only for that original antigen. So all of these have a little triangle shape, none of them are circle. So we selected this one B cell, clonal selection, and only it's being selected for the triangle shape, okay, or the little diamond, that's a diamond, okay? It's selected, we're using that one. Of all the millions of B cells you have, we selected this one because it bound to this antigen, that's clonal selection. Okay. Then it presents it to a T helper cell. T helper cell via the T cell receptor sends cytokines that activate a B cell. Now we're going to expand, we're gonna make a lot of copies of this B lymphocyte and its plasma cells so we can make enough antibodies to combat the infection. Okay. So clonal selection is, we're going to take this B cell binds only this antibody and make plasma cells and memory cells that are specific only for that antigen that it encountered, okay? What can happen is sometimes things go wrong where you have B cells that will attack self, B cells and T cells. So you have to be able to eliminate those from your body. If you don't, that's how you get all of your autoimmune diseases. Your body is recognizing you as the antigen that it needs to destroy. So autoimmune diseases come from B lymphocytes and T lymphocytes making it through that shouldn't. Okay, putting it together. So at the top we have a pluripotent stem cell. Stem cells differentiate into B cells. Well, B cells will mature in the bone marrow, okay? They're circulating through the bloodstream. On the surface, they have the IgD, the B cell receptor, circulating through their bloodstream until they come and encounter an antigen that it is specific for. Once it, that scared me. I was like, oh God. I taught over 10 years at Stockton, not one fire alarm, except the one that I caused. And then here we're like 4. I'm like, we blew up something in Oak. I did. My lab partner behind me, his whole round bottom flask exploded. As soon as he exploded, he took his goggles off, and the glass and everything went in his eyes. Like 2 weeks after 9/11.
Is that the banana guy?
Oh, the banana guy, yeah. I always remember him, right? He's probably a chemist somewhere now. I'm famous, writing papers. Okay, so when we have the B cell data selected, okay, then it's going to differentiate into the plasma cell or to the memory cell. Are we good on this? Because I'm trying to read eyes. Overwhelmed? Making sense? Making sense?
A little overwhelmed.
Overwhelmed. Okay, ask me questions, cause I'm reading your faces, I promise you I am.
So everything is starting as just a general cell, and then it's gonna differentiate and branch off.
So,
And that's as far as.
a B lymphocyte, so you have, a B lymphocyte can differentiate into a memory B cell or a plasma B cell. That says, okay, I know what the antigen looks like. Now I can make a memory cell to save for later, the next time I encounter this. And I'm gonna make a plasma cell for the right now infection and make a bazillion antibodies that can now circulate through the bloodstream, bind to that pathogen, and then neutralize it so it can't continue to replicate and cause further infection.
So the plasma, you would find those in the Golgi and the rough ER,
There's Golgi and rough ER, those are organelles.
right?
So when you're seeing this different, you see how the shape is a little bit different here on the plasma cell, elongated versus circular here? There's a lot more rough ER and Golgi. Here the cell says, I'm gonna need to make, just like if you're like, think the Amazon shipping center, right? Okay, I used that as an example as a Golgi before. I'm gonna have to ship a lot of products, okay? I'm gonna have a distribution center like Amazon out here, okay? So on plasma cells, they have an abundance of Golgi and rough ER inside of them because their one job is to make a ton of protein, rough ER ribosomes, and I have to secrete all those proteins, Golgi apparatus' job, to get them out of the cell and into the bloodstream, okay?
And then the memory cell, what's in the memory cell?
The memory cell says, I know how to, so let's say, second time I get infected, okay, so I go through mononucleosis once, done. Come back around, someone else has mono. My memory cells will automatically turn into plasma cells. They don't have to go through all of this again. It's like I made a copy, and anytime I come around again, I can automatically activate the memory cell, and will go straight to a plasma cell and make antibodies again.
That's secondary response?
That's secondary response. And we'll get into it in a few slides, I was mentioning now since we're here. So in a primary response, we make IgMs. On the secondary response, we'll make IgGs.
So when you have the antigen presenting cell, that basically like, okay, here's like facial recognition of what we're looking for.
Okay, any other questions? No questions to be like, what's an antibody? Okay, what's an antigen? You can ask those questions. What class am I in? Okay, why did I become a nursing major? Okay, those are all fair right now. Okay. So we can classify antigens into two types. Okay, those that are going to require help of a T cell. Okay, an antigen presenting cell has to show it off to a T helper cell, just like the slide we saw before. Or you can have a T independent antigen. It doesn't need to present to a T helper cell to make antibodies. Okay. The ones that say, hey, my antigen is my B cell, here's my antigen, let me show you off to a T helper cell. They have that interaction between the two, and the T helper cell activates the B cell to make antibodies. They're gonna have a stronger response, and they'll create the memory cells and the plasma cells, and so you'll have a longer response and be ready for a secondary response as well. Okay. T-independent antigens do not create memory cells, so you're gonna have a weaker response and you don't have a long-term response. Okay, so examples of T-independent antigens are things that are long polysaccharides. They have highly repetitive units, so like sugar, 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 glucose, 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 over and over and over again. Those are the ones that will not present to a T helper cell and thus don't get a long-term response. Okay, so there are different types of interactions, and once, let's say our plasma cells making all these antibodies, okay? There's different types of reactions that they can have with antigen. They bind to that antigen, but there's different applications. Okay. The affinity for an antibody to an antigen is called, that's affinity. How close is the bond there? Okay, how close is the interaction between the amino acids of the antigen and the amino acids of antibody. Okay. So once they bind to that antigen, you can have an example, agglutination. Okay. So agglutination is clumping. Okay, I have all these antigens. Let's say I have 5 E. coli cells around me. Okay, and I'm an IgM antibody. Remember, I come in 5, right? So I have 10 spots I can bind to. So I can bind this cell over here, I can bind that cell over there, use my leg, bind this one over here, use my leg to bind something else, right? And my mouth. There's 5 spots, 5 antibodies. And so I clump all these cells and keep them from spreading, and then your body can destroy those 5 E. coli cells because the antibodies tag them and they can't get into your intestinal tract. They can't burrow in. It's like, if you're a mom, you know how you get your kids from running across the street, like it's instant, right? Response. Your kid snatches you, you're like, got it. Okay, that kid's not gonna run out because you tagged it. You know, we got 3 kids. You become like a superhero because you can do it with your leg and your pinky finger, and you grab all 3 and you're holding the door, right? Because they can't spread and go and cause infection or get into trouble somewhere else. Okay, opsonization is when antibodies are coating a bacteria and they attract phagocytes to that particular bacteria. Okay, got some pictures for you, let's show you. Okay, so agglutination, our top picture here, you have antibodies that are binding multiple microbes, and so you're clumping them together. Opsonization, antibodies completely coating a microbe, so they're binding to antigens on the surface of that cell, and that promotes phagocytosis. So a phagocytic cell, a macrophage, or a dendritic cell will come in and go. Okay. Neutralization is when you have an antibody, say, binding to a bacteria or to a virus, okay? Just that one, so it's not clumping, it's not agglutination, we're not binding multiple bacteria, okay, just binding one, and it prevents it from binding to, so let's say this is Shigella or Salmonella, and I'm coating all the antigen sites, and so now Salmonella or Shigella can't bind to my intestinal mucosa, let's say it's IgA, can't bind and cause membrane ruffling and can't get inside. Okay. Or I can bind, let's say, the HIV virus or the influenza virus, so it can't bind to specific receptors, let's say in my pseudostratified columnar epithelium in my trachea. Okay, so it's a blocking mechanism. I brought this up a few slides ago. Fancy long name for a pretty simple process. Okay, so antibody dependent cell mediated cytotoxicity. Long name, okay, ADCC. Let's break it down. What does that mean? So here we have a really large parasitic worm, and a phagocytic cell like a macrophage just can't come and swallow up a whole worm. It needs help, okay? So the way we can destroy this worm is on the surface of the worm, we're gonna have antigens, okay, from its cell wall, its cell membrane, whatever it might be. So what happens is IgE will bind to the antigen. So this is our variable region, binds to the antigen. Okay. And then the FC region of the antibody binds to a receptor on a eosinophil. Okay. And then that eosinophil releases what's called perforins and granzymes. Perforin will perforate. Granzymes, they just come, they're the hole punchers. I've used that term a few times. They come and just wreck the cell membrane and cause it to lyse open. Perforins, P-E-R-F-O-R-I-N-S, perforins, and granzymes, G-R-A-N-Z-Y-M-E-S. So those eosinophils will release those enzymes. They're enzymes that will lyse open this worm. So imagine you're doing that all across the surface of the worm. Multiple IgEs are bound, and they're binding to multiple eosinophils. All those eosinophils at once will release the granzymes and perforins and completely obliterate the parasitic worm. Okay. So now we're going to kind of focus on T cells for a second. So T cells, we talked about so back it up. B cells produce antibodies. Are we good there? Okay. The B cells produce antibodies that target antigens that are outside of cells. Okay. They can do that through agglutination. They can do that through opsonization, neutralization, antibody-dependent cell-mediated cytotoxicity. Say that 10 times, right?
They
Yeah.
have cells outside as well.
They attack antigens that are outside of the cell. Or on top of the cell, but it's an exterior antigen. Then we have T cells, which is part of cell-mediated immunity. These are going to target cells that have had struggles with an intracellular infection. So viruses replicating inside. Chlamydia, one of those type of guys that's an intracellular parasite or bacteria. So once T cells or T lymphocytes leave the bone marrow, they enter into the thymus where they finish the maturation process. Okay? Then those T cells, if they're good to go, they'll mature and leave and enter the lymph nodes, spleen, wherever they need to go. If they recognize you, they are deleted, okay? It's called thymic selection. Those are cells that like, hey, do you pass the test? Can you keep going in this body? Great. Are you gonna react towards me? Then you have to be destroyed. So that's where T cells go through their selection process, where they should go on or they need to be destroyed. Okay. So once they pass the clearance and they're not gonna react to us, then they can go to lymph nodes, spleen, Peyer's patches, tonsils, all the lymphatic tissue throughout our body to be ready to encounter antigens. Okay. The way they bind to antigens, they don't have antibodies. They don't have a B cell receptor. They have a T cell receptor. It's not an antibody, it's not IgD, it's different proteins involved. So now we're gonna look at a T cell receptor setup. Okay. I wanna go to, let me come back to this in just a second, okay? Keep going. I'm gonna come back to these in just a second. Antigens and cells. Trying to, we'll stay here. Okay. I'll try to look at my time. So, when we look at antigen presenting cells going to take a little piece of antigen and present it to a T cell, okay, a T helper cell. So one way that we do this, if one of the things that we're going to constantly encounter pathogens are our GI tract, eating food that's not good, eating things that went, the chicken that went south, the cheese that looks funky, the milk that went bad, whatever it might be. So when we take food in, encounter a potential E. coli or a Campylobacter, and it gets all the way through, walk through your GI tract, we're gonna help Nyla out. But first thing, after oral cavity, what do you go through? Do you remember what your tube's called again?
Esophagus.
Esophagus, okay. Esophagus to your stomach. Where do you go after that?
To the intestines.
Which one, small or large? Small. Okay, then we go to our large. So we go duodenum, jejunum, ileum, and then through your colon, large intestine. So in your ileum, that's where you have your Peyer's patches at, okay? So in your Peyer's patches, okay, when you guys did your histology and anatomy and you looked at the ileum, it looked like Easter eggs, so these round ovals in the intestine slide, okay? So in that Peyer's patch, you have M cells. Those M cells are lymphatic cells that when a bacteria invades your mucosa, like Salmonella, Shigella, E. coli, all these guys, it comes through. It's going to be processed via an antigen presenting cell, a macrophage, and then that macrophage will present it to a T helper cell. Other antigen presenting cells we have are dendritic cells. We haven't talked a lot about those. Dendritic cells, we also have macrophages. These are cells that can engulf bacteria, phagocytose them, and present the antigen on the surface via an MHC2 receptor. So dendritic cells, they're going to engulf it, they're phagocytic. Okay? You find them a lot in your skin, in your dermal region. They're going to be in your genital tract as well, and you'll find them in your thymus in your blood. The second one is your macrophages. Macrophages are monocytes that left the bloodstream, entered the tissues, and they're there waiting. We have free macrophages that are just moving around, and fixed macrophages. There are two categories. They will go through phagocytosis, okay, and then they'll bring that in, take a little piece of it, and present it on an MHC2 receptor. Okay, here's an example of a dendritic cell, okay? Main cell body, and all these extensions are what will reach out to engulf a bacteria and bring it in. And here's the two macrophages, the difference between a resting one that has not encountered an antigen or went through phagocytosis versus a second one that has went through phagocytosis and then has been activated by cytokines. So, I'm an antigen presenting cell, macrophage, dendritic cell, one of those guys. Okay, I can take my antigen, I'm gonna show it off. Okay, we looked at how B cells did it, right? Now we're looking at macrophages and dendritic cells as well. They're gonna show it to a T helper cell or to a second type called a cytotoxic T cell. We have two cell types, cytotoxic T cells and helper T cells. Okay. Our T helper cells are also called CD4 cells. Our cytotoxic T lymphocytes are called CD8 cells. I'll tell you why that in just a second. T helper cells will recognize antigen that's presented by MHC2 receptors. Okay? I want you to highlight and underline this. You gotta get this nailed in your head. MHC2 receptors are only on antigen presenting cells. That could be a B cell or a classic APC such as a dendritic cell or a macrophage. So B cells, macrophages, dendritic cells, they have MHC2 receptors. So an antigen presenting cell that has MHC2 molecules will say, hey, I'm gonna present my antigen to only a T helper cell. Yes, ma'am.
You said B cells only attach to MHC2?
B cells have MHC2 receptors on their surface. Let me take you back real quick. This slide. B cell had a B cell receptor, right? Bind that antigen, take it in. This little dude right here, that little yellow semicircle, that's an MHC2 receptor. And it presents the antigen on that receptor to a T helper cell. Presents it, this is a T cell receptor in bright orange, the antigen, and MHC2. Okay, so I'm gonna say it again because I'm looking at your eyeballs. Okay. Yellow's MHC2. Little triangle's the antigen. Bright orange T cell receptor. Okay. And so B cells have MHC2 receptors and they present to T helper cells. Okay, so T helper cells are going to bind antigen on MHC2s. Okay, CD8 cells, cytotoxic lymphocytes, their T cell receptors can bind to cells that have MHC1s. So first thing I said earlier in the lecture, all of our self cells, everything except an erythrocyte has an MHC1. So they can interact with anything that's a self cell except an erythrocyte. So how this works is, let's work with our first example, the slide I just showed you. So I have a B cell, bound to an antigen, I internalized it, and I stuck it on my MHC2, right? I showed it to my T helper cell. T helper cell activated me. Great. Okay. I made memory cells and I made plasma cells. The T cell receptor on the TH cell recognizes and binds the antigen on an MHC2. Okay. Once that T helper cell gets activated, it tells the B cell, go make memory cells, go make plasma cells. Great, you do your job. Okay. I'm also gonna do my job. I'm gonna differentiate into 3 different subsets. I can differentiate into a T helper 1. I can differentiate into a T helper 2, T helper 17, and I will make my own memory cell as well for the next time I have to fight the same pathogen. So the 3 subsets that are made, we need to do the 3 subsets, and I'll go back to that figure. So T helper 17s, they release a protein or a cytokine called interleukin 7. The IL stands for interleukin 17, sorry. That's gonna cause inflammation to happen. So your T helper says like, hey, we're fighting a serious pathogen, we need to shut it down. I'm gonna cause the redness to happen, the pain to happen, the swelling, the heat, to try to contain this pathogen right here and make it be phagocytosed. We have T helper 1 cells. They're going to produce interferon gamma. Highlight the gamma because different function than alpha and beta. Okay, that can activate macrophages that come over here and phagocytose all of the cells, all the pathogens. Okay, it can enhance complement. Okay, so opsonization can be activated. Okay, and it promotes antibody production. So there's a lot of functions that can help from TH1 being activated. TH2, that releases interleukin 4. Okay, that's going to activate B cells to make IgE and will activate eosinophils. So if I'm having a bacterial infection, I'm likely not going to have TH2 produced. If I'm having a parasitic infection, worms, Giardia, something like that, I will have TH2 produced. If I'm having a bacterial infection, it's going to be TH1 cells that will be produced. Let me go back to this figure here. Okay, so T helper cells recognize antigen on MHC2s. MHC2s are on antigen presenting cells. Macrophages, dendritic cells, B cells. So I want to say it over and over again. So an APC is a macrophage, a dendritic cell, and a B cell. So here we have a dendritic cell. It's engulfing an antigen on a bacteria. Okay. We go through phagocytosis. We're gonna break up our antigen. It's gonna couple with an MHC2 receptor. Both of those will get put on the surface of the cell. They're gonna be presented. Okay. The MHC2 will show off the antigen to the T helper cell. The figure we did in the beginning of the lecture, this was a B cell with an antibody. Now we're looking at a dendritic cell doing the same exact thing. If it was a B cell, this would end up in memory cells and plasma cells being made. It's a dendritic cell we present to a T helper cell, then that can differentiate into a TH1, a TH2, or a TH17. Okay, we talked about the final effects. I'm going to scoot for time. Okay. As another subset of T cells is T regulatory cells. They're a subset of T helpers or CD4s. Okay. One thing I think that will help you visualize when I say CD4, let me do this on the board real quick. Here's my cell. This will be my T cell receptor. And I'm going to interact with an antigen presenting cell. So because I'm doing an interact with an antigen presenting cell, what 3 cells could I be?
B cell.
B cell, dendritic, macrophage. Okay. Which MHC receptor will I have? 2, right? MHC2. Okay. So if I'm an antigen presenting cell and I'm an MHC2 receptor, I'm sorry guys, helper. I'm going to be a helper T cell. Okay. A helper T cell is also a CD4 cell. So what the heck is a CD4? So a CD4, CD stands for cluster of differentiation. Okay. There's a T cell receptor and an MHC2 receptor. They both bind to the antigen. So that's only antigen. What actually does the recognition that this is an antigen presenting cell, this is MHC2, is something called the CD4 receptor. That is right next to it, and it binds to the actual MHC2. So there's two interactions. This is where this textbook is lacking on some detail in the figures. Okay. So T cell and MHC2 directly bind the antigen. The CD4 only touches the MHC2. Okay. If I am a cytotoxic T cell, okay, I'm going to have my T cell receptor. Okay, I'm going to bind to a cell type. Okay, what cells am I doing antigen presenting cells? No. Okay. So I'm going to be doing self cells. What type of MHC do I have? One or two?
One.
One, right? So MHC 1. They both bind to the antigen. Okay. But a cytotoxic T cell has, it's a CD8, cluster differentiate CD8. That is a co-receptor that binds only to the MHC. Does that make more sense, those two drawings? Okay. So, we have two types. Helper T cells bind to APCs with MHC2s. The CD4, so it's a you can think that CD4 and CD8 are co-receptors. So you have two interactions happening, the co-receptor and the T cell receptor. So on multiple choice questions, long answer, whatever, you have to be able to, if I say T helper cell, I need you to know CD4. If I say cytotoxic, I need you to think CD8. Okay, if I say CD8, cytotoxic, you have to get pretty comfortable interchanging those two. T cell receptor. Mm-hmm.
Why do they have to attach to the MHCs?
Yeah, so this recognizes just antigen, this recognizes which type of MHC it is.
So they can.
So this one only binds to ones, the 4 only binds to 2s. So this T cell says, I got the antigen, you take care of making sure this is the right MHC. So both are confirming what cell type they're bound to.
Isn't there one of them that like kills or something, like the helper cell doesn't, but
The cytotoxic T cells, yeah, that's where we're going right now. Yep. It'll directly kill cells, exactly. So if I do this version here, okay, my T helper cell is now activated. I can tell if I'm a B cell, man, where's my freaking marker? They're all dead. These guys. So if I'm a B cell, okay, I'm going to activate this. What's going to happen to the B cell? Plasma and memory. Right? The helper T cell is also activated. And I can make TH1s, TH2s, TH17s. All right, so now we're gonna add the next layer onto this. Another subset, which is the slide that's on here, is a T regulatory cell. Okay, being able to not attack self, right? Don't attack my own body. If I do, I've got autoimmune disease. How the heck do women carry babies? With a whole different DNA, genetics, whole different makeup, and not attack it. Okay, why are there spontaneous abortions happening all the time? Okay, it's because of these guys. T regulatory cells protects your baby, and it protects your normal flora. So if I have all this bacteria in me as well, why is my body like saying, hey, regular E. coli, go bye-bye, regular Enterobacter, okay? So all of your normal flora is able to survive, and your baby will survive because of this particular cell subset. All right, now we're gonna focus on our cytotoxic T cells. Okay. So cytotoxic T cells start off as a precursor CTL, or cytotoxic T cell, and they become mature, they get activated. Okay. So the picture on the board back here. Okay. Cytotoxic T cells can be activated by T helper cells. So that arrow that I drew back there, Th1s, 2, 17s, regulatory, I could also draw that arrow all the way over here and activate cytotoxic T cells. Okay, so I want to take you to our image here, walk through this. So I have a cell that has been infected by a virus. Okay. So inside, lytic cycle, lysogenic cycle, all that's happening inside this particular cell. So this cell says, okay, I'm going to take, it's a self cell. Respiratory, so we'll stick with respiratory cells. It's a respiratory cell. All self cells have MHC1s. Okay? Those are like, crap, I've got a virus. Okay, I'm going to take a little piece of that virus, an antigen, and present it on my MHC1. I'm going to put it there. So now we have what's called processed antigen being shown off on MHC1. Okay, that's going to bind to a T helper 1 cell. Okay, the T helper 1 cell will activate a precursor cytotoxic T lymphocyte, and then once we have the activated cytotoxic T lymphocyte, that secretes those enzymes, you've heard about them again, perforins and granzymes, and that will cause or initiate apoptosis or apoptosis, have you heard that, in a cell. Okay, so let's walk through apoptosis. I've got to scoot for the time. Apoptosis is called programmed cell death. So it's not like just a bomb landed on a cell, it exploded, okay? It initiates apoptosis where everything starts getting digested, all the organelles, cytoskeleton, all that starts getting destroyed inside the cell. That way waste products are not just blown into the universe around the cell. Okay, so when we look at an apoptotic cell, we are going to get what's called this blebbing. Okay, so blebbing, all these little protrusions here is a cell slowly destroying itself. So if this is a viral infected cell and I destroy everything inside, okay, to keep it in-house, then I prevent that virus from spreading. If I just throw a bomb at it and let it all lyse open, I could potentially release viruses to neighboring cells. So programmed cell death keeps the virus in-house, shuts that cell down section by section. Okay, our next lymphocyte or out of the lymphoid stem cell line is our natural killer cell. Okay? So natural killer cells are granular leukocytes, but they don't express MHC1 antigen, so they're not going to present antigens on the surface. Okay, they will target, they're unique for virus-infected cells, and they will also target tumor cells, cells that have been taken over by a virus and become cancerous because of it. Okay? So they don't always have to have an antigen present to become activated. And these guys will also secrete granzymes and perforins that can initiate apoptosis for viral infected cells. This chart is a very good chart, just a very good summary chart on the focus of each subset of T helper cells. So I want you to know this, get this guy, it's a very good one. So now let's just wrap up with the, so not a bacteria, not a fungus, not a virus, I've got huge parasites or maybe protozoas that I can't digest. Okay, I can't phagocytose them with a dendritic cell or a macrophage. So then this is where we have to use the antibody-dependent cell-mediated cytotoxicity step. So we take this particular microbe or microparasite, all of the antigens on the surface. I talked about IgE. The variable region binds to the antigen, but the Fc region binds to receptors on eosinophils or macrophages. Then those cells will then release enzymes that can come and destroy the worm. So you have an interaction of multiple IgEs, multiple eosinophils, all taking down the worm. First response, okay, short-lived, IgM's the first one out. Secondary response, okay, I have memory cells now. Memory cells remember what I need to do. I don't have to go through presentation and activation anymore. Memory cells can go straight to B cells. Okay. So what happens is I can do what's class switching. I don't need to maybe use IgM anymore. I can use IgG or IgA. I can activate a different type of antibody. So usually it's going to be IgG that comes out for the secondary response. Okay. So if I want to know if you've been exposed to something, okay, I can take your titer, okay, and depending if you have IgM, I know you've only encountered it once. If you have IgGs for a specific antibody, that means you've got long-term immunity. You've been vaccinated. You've had a couple exposures to this particular one. So the variable region is the same. I want to make sure I've got that clear. The variable region is the same for the same epitope, same antigen. It's the bottom portion, the FC, that switches. IgM for the first time, IgG for the second time. Which one you have in your blood tells me if you're on a primary response or a secondary response. Vaccination or a booster, I can tell. So when we were putting these together, let's say first time I've encountered mononucleosis. Okay. IgM is going to be my fourth after a few days of prodromal period and symptoms are starting to settle in. Okay, IgM is going to be the first one out. You will have IgG produced, but it comes later. Okay, on your secondary response, the first guy out of the gate is your IgG, green, much higher response, much more antibody made. You do have some IgM made, but it's at a much lower level. Okay, so they're both made. It's just IgM's higher first out of the gate on primary. Secondary response, IgG is out of the gate first in a much higher titer. Okay, last slide. So the types of immunity that you can have. Let's look at the chart. You have natural immunity and artificial immunity. So under the heading of adaptive immunity, this whole chapter, you can have naturally acquired or artificially acquired. Naturally acquired means I've had this infection. I got sick. My body, my B cells made antibodies against this because I'm the one who had mono, okay? Passive means I have antibodies because my mom gave them to me via the placenta, via breast milk. I didn't have to encounter this antigen to have these antibodies. They were just given to me. Artificially acquired, artificial means you didn't encounter you didn't get sick, you had a vaccination. We physically put the antigen inside of you. You made antibodies to it, but it's because of vaccination. Artificially acquired passive is when you get what's called an immunoglobulin treatment. You get antibodies injected into you. So my mom gets this for her MS flare-ups. They give her antibodies that come and bind all of her. So 30 seconds. MS is an autoimmune disease, right? My mom's antibodies, they attack all of her axons that are unmyelinated. So she has all these flare-ups because all the antibodies are attacking. Okay. So she goes in when she has a flare-up for an immunoglobulin treatment. All these antibodies come and it's cool. They bind to her antibodies and say, hey, stay away. They treat them like the antigen. Bind you up so you can't bind her axons. Someone told me the price of this one time. It's like $20,000 or $30,000.
A treatment.
A treatment. I was like, I know how many bags we get hung when we go in. I was like, thank you for insurance, because, yeah, pretty sure we knock out $60,000 every time we go in for treatments, depending on how bad it is.
That's what my father-in-law has to do the
IgM treatment, yeah. So, let's do, naturally acquired, I got sick. Natural passive, my mom gave it to me. Okay. Artificially acquired active, vaccination. Artificially acquired passive, I'm getting an immunoglobulin therapy in a hospital setting usually. Okay. All right, where are we at on stress level? Because I'm reading all your faces today, and they are nowhere as excited as they were with one.
975.
Everybody looks like over it. Over it.
Just 1 more